फैसलाबाद। अब हम जो है वो एलिजिबिलिटी क्राइटेरिया की बात करते हैं ईस्टर्न मेडिसिन में एडमिशन्स के लिए, जो कि एफएससी है, और मुख्तलिफ़ जगहों पे इसका जो है वो मेरिट बहुत ज़्यादा हाई है, जिसमें GCUF के अंदर जो है वो 80 तक तकरीबन जो है वो है। और इस GCUF के अंदर जो है वो 2 शिफ्टें चलती हैं, एक जो मॉर्निंग की और 7 रेप्लिका। उस साथ जो है वो एमफिल और PhD प्रोग्राम भी जारी है। और सबसे ज़्यादा तादाद रखने वाला GCUF है, जिसके अंदर जो है वो स्टूडेंट्स की तादाद पाकिस्तान में सबसे ज़्यादा है और बहुत बड़ी तेज़ी से जो है वो ग्रो कर रहे हैं। वो बहुत कम मुद्दत के अंदर इस डिपार्टमेंट ने ग्रो किया है। और अगर हम ये स्टडी के लिहाज से देखा जाए तो स्टडी जो है वो तकरीबन हम लोग जो करवा रहे हैं, तमाम मेडिकल साइंसेस की जो है वो स्टडी हम करवाते हैं। इसके अंदर जो है वो अहम सब्जेक्ट जो मैं पढ़ाता हूँ वो है आपका जो है वो बायोकेमिस्ट्री। बायोकेमिस्ट्री जो है, इसमें अगर देखा जाए तो बायोकेमिस्ट्री में आज हम डिस्कस करेंगे ग्लाइकोजन स्टोरेज डिजीज के बारे में। तो ग्लाइकोजेनेसिस क्या है? बेसिकली इट इज़ द एक्सेसिव glycogenosis. Basically it is the excessive accumulation of normal or abnormal glycogen. There are a group of genetic diseases that results from a defect in an enzyme required for either glycogen synthesis or degradation. Deficiency of these enzymes results in the accumulation of excess amount of glycogens whose structure is either normal or abnormal. Deficiency or defect may be in a single tissue like liver, or it may be generalized, affecting muscle, kidney, intestine, and myocardium. General effects may range from fatal in infancy to mild disorders that are not life-threatening. There are 6 types of glycogen storage diseases. Type 1, Von Gerke disease. Type 2, Pompe disease. Type 3, Cory's disease. Type 4, Anderson disease. Type 5, McCordle disease. Type 6, Hertz disease. Von Gerke disease. Type 1, glycogen storage disease. Deficiency of glucose 6-phosphate phosphatase. Increased glycogen stores. Normal glycogen structures affects liver, kidney, and intestine. Severe fasting hypoglycemia, fatty liver, and hepatomegaly. Pompe's disease, that is called type 2 glycogen storage disease. Deficiency of lysosomal alpha 1,4 glycosidase, acid maltase. Normal glycogen structure, normal blood glucose level. It is a generalized disease affecting liver, heart, and muscles. Excessive glycogen concentrations are found in abnormal vacuoles in the cytosol. Severe cardiomegaly. Early death usually occurs. Cory's disease type 3, glycogen storage disease. It is also known as limit dextrinosis and is due to deficiency of the branching enzyme. There will be excess accumulations of polysaccharides of the limit dextrin type. Anderson disease type 4, glycogen storage disease. It is also known as amylopectinosis and is due to the deficiency of branching enzyme. Causes accumulation of glycogens having few branch points. Myocardial disease. Type 5, glycogen storage disease. Deficiency of muscle glycogen phosphatase. Skeletal muscles are affected. Increased stores of glycogen in muscles with normal structures. Liver enzymes are normal. Normal mental development. After exercise, temporary weakness and cramping of muscles. Normally, lactate rises during strenuous exercise. In this type 5 glycogen storage disease, there will be no rise in blood lactate during strenuous exercise. House disease, type 6 glycogen storage disease, deficiency of phosphoglucomutase in the liver. Hexose, now we will discuss the hexose monophosphate shunt. Other names of are pentose phosphate, R. Phosphogluconate pathway. The importance of high HMP pathway is mainly due to NADPH2 produced in this pathway. NADPH2 is the major and the most important biochemical reductant. There is no exact definitions of the pathway because it produces many sugar intermediate for different carbohydrate metabolism metabolic processes, and unlike glycolysis, it is multi-directional. It consists of two irreversible oxidative reactions followed by series of sugar phosphate interconversions. It occurs in the cytosol of erythrocytes, liver, adipose tissues, lactating mammary glands, thyroid, adrenal cortex, testes, and ovaries. Some important points about the steps of HMP shunts. No need to memorize the steps. Glucose to glucose 6 phosphate is not the first reactions. Neither it is a part of HMP shunt. Rather, it is a common reactions for both glycolysis and HMP shunt. HMP pathway starts with glucose 6 phosphate. HMP shunt is also called direct oxidative pathway because oxidation occurs in the first reactions glucose 6 phosphate to 6 phosphogluconolactone with the production of 1 NADPH2 of the pathway. Second, NADPH2 is produced by oxidative decarboxylation of 6 phosphogluconolactone with lac- lactone with the release of carbon dioxide. This carbon dioxide is a characteristic product of HMP shunt. Rate limiting enzymes. Glucose 6 phosphate dehydrogenase is the key enzyme. The enzyme produces 1 NADPH2 of the pathway regulation. The enzyme glucose 6 phosphate dehydrogenase is regulated by NADP over NADPH2 ratio. Increased NADP NADPH2 ratio stimulates the pathway to produce more NADPH2. Decreased NADP over NADPH2 ratio inhibits the pathway. Importance of HMP pathway. In this pathway, not a single ATP is directly consumed or produced. It is the only source by which body gets reduced form of NADP. For example, NADPH2. Two molecules of NADPH2 are produced for each molecule of glucose 6 phosphate entering the oxidative part of the pathway. Pathway maintains the integrity of RBC by producing NADPH2. Pathway produces pentoses, ribose, which are required for the biosynthesis of nucleotide, RNA, and DNA. It produces intermediate products for glycolysis. Uses of NADPH2. NADPH2 is a biochemical reductant and is the reduced form of NADP coenzymes. NADP is a derivative of niacins, water-soluble vitamins. NADPH2 helps in the biosynthesis of fatty acids and cholesterol. It causes reduction of hydrogen peroxide and superoxide. Hydrogen peroxide and superoxide are oxygen-derived free radicals which are highly reactive and can cause serious damage to RBC, DNA, proteins, and unsaturated fatty acid. It reduces glutathione of RBC. Glutathione is a polypeptide present on the cell membrane of RBC, which on oxidation makes the RBC fragile. So, in order to survive and maintain the integrity of RBC, it is very important to keep the glutathione in a reduced form. NADPH2 is required by the liver microsomal cytochrome P450 monooxygenase cytochrome P450 system for the hydroxylation of steroid, hormone, alcohol, and many drugs. NADPH2 is also required by RBC for phagocytosis and killing of bacteria. Comparison between glycogen and HMP shunt. Glycogen. Clinical notes: glucose 6 phosphate dehydrogenase deficiency. It is an inherited X-linked disease. Therefore, only males are affected and females are only carrier. It is very rare for females to have glucose 6 phosphate deficiency. It is glucose 6 phosphate dehydrogenase deficiency. It is the most common disease producing enzyme deficiency. Deficiency of glucose 6 phosphate dehydrogenase results in deficiency of NADPH2. Deficiency of NADPH2 increases the concentration of oxidized glutathione. Oxidized glutathione is unable to keep the cell membrane of RBC in reduced form, hence RBC become fragile. RBC become weak and vulnerable to oxidative damage by hydrogen peroxide, superoxide, and oxygen-derived free radicals, resulting in hemolysis. Glucose 6-phosphate dehydrogenase deficiency is characterized by hemolytic anemia only when a person is exposed to oxidizing agents, hydrogen peroxide, and superoxide. Interestingly, patients with glucose 6-phosphate dehydrogenase deficiencies are resistant against falciparum or malaria. Precipitating factors, oxidate drugs, 3 A's, antibiotics, anti-malaria, antipyretics. Thank you very much.